Hi everyone, my name is Jacob Nearing, and today I'll be presenting to you my poster on preserving and assimilating region-specific ambiguities in taxonomic hierarchical assignment for amplicons, or for short, PARATHA. As many of us know, short-read amplicon sequencing is a common strategy to taxonomically profile microbial communities, and this generally involves the application of a specific gene or region within the genome, such as the 16S ribosomal RNA gene. And while this is a fairly low-cost strategy, it does suffer from some drawbacks. One being that the usage of different variable regions can result in the differential identification of taxa, as well as some short variable regions not always offering or being able to identify between different taxa, which introduces further ambiguity. And so what we wanted to do with PARATHA is directly model ambiguity of taxonomic assignment to improve the assignment of amplicon sequences, whether or not that be 16S sequences, or for people that are interested in fungal communities, they might be using ITS sequencing, or perhaps even 18S sequencing. And so here we have the general overview of the PARATHA workflow. And so what it does is it takes in a database of sequences. So in this case, we're using a Silvus seed database of about 7,000 sequences. It takes in a primer sequence, so the primers that you may have used in your study, and then it takes in the taxonomy of those sequences. And so what it does, the first step is it takes those sequences and it trims them down to the specific primer region that would have been amplified in your experiment. It then takes those trimmed sequences and puts them into FastTree to generate a primer-specific phylogenetic tree. And so the hope is that this tree models some of the ambiguity that would be seen within the variable regions or the region that you sequenced with the primers that you used. We then take this phylogenetic tree, and using a distance-based algorithm, we assign taxonomy to the internal nodes of the tree. Once we have the internal nodes labeled, we can then take query sequences, so those sequences that you might have sequenced in your own experiment. We can place them into the tree using pplacer, and using the best placements of those query reads, we can assign taxonomy to them. And that is the general workflow of how PARATHA works. I'm going to spend a little more time talking about what I mean by this distance-based algorithm for the taxonomic assignment of internal nodes. And so this occurs in two steps, the first being the identification of an optimal taxonomic distance threshold. And so what we can see here is that we have a phylogenetic tree, such as this one, where we have at the tips labels A, B, C, and D. And in this case, what we can do is we can select different distances, phylogenetic distances, to cut the tree up to identify different groupings of the labels. And so we can see if we chose distance 1, we'd end up with two groupings, one grouping A and one grouping B, C, or D at this node here, or this split point here, I guess is what I should say. And so we can then measure the error rate of these groups, where we can see that we've overgrouped by 2. We've basically grouped two extra sequences. We have grouped two extra sequences together, but we do have an oversplitting of 0. We can also choose much shorter distances, such as distance 3, where we might end up oversplitting our A into 3 separate groups, generating 2 additional groups or splitting errors, but then we don't overgroup B, C, or D at all, and so we get an error of 0 for overgrouping. Finally, we can pick a distance of 2 where we have A all by itself, B all by itself, but then a overgrouping error of C or D. And so what we can do is we can look at the table and calculate the total error rate of each distance, and we can see that in general distance 2 has the lowest error rate, and so it's most likely the optimal distance for this taxonomy. And so what we can do is we can do this across different taxonomic levels. So in this case, we can see at the phylum level, the distance threshold is generally fairly high, whereas at the species level, it's fairly low. And we can even do this across different variable regions. So in this case, we've done it against V1, V2 region of the 16S RNA, ribosomal RNA gene, or the V4, V5, 16S ribosomal RNA gene. And so once we have the optimal distances, we can then use those distances to start assigning labels to the internal nodes of the tree. So you can imagine we have the same tree as we did last time here. And so anything above the distance threshold that we've chosen remains unassigned at that level, whereas anything below it will get an assignment. And so in this case, it's pretty easy to assign the label for this internal node because it's A, because everything below it is A. Similarly, we can look at B and we can see everything underneath this label is also B, so we can assign it B. Although it becomes a little more tricky at this node, and that's because if we look under this node, we have both labels C and D. And so what we can say is that this internal node is actually ambiguous. We can't actually determine the correct taxonomy for potentially a sequence that's placed there because it could be either C or D. And so what we do is we label this internal node as C or D. And in cases where there are multiple different groupings, we have ways of dealing with potential error in the annotation process using a binomial error model. If you have more questions about that, I'd be happy to talk about it. Finally, once we have a tree that has its internal nodes labeled, we can then assign taxonomy to new query sequences. So in this case, you can imagine that this orange sequence is a sequence from our study that we placed into our tree. This orange sequence gets both a pendental length and a distal length. We can then calculate the distance from this new place sequence to all of the children's sequence underneath its index node here, this index node here. And we can see if this distance is within the species level. If it's within the species level threshold, then we can assign the species taxonomy to this. And we do this at each taxonomic level. So you can imagine potentially maybe the sequence is within the family distance, but not within the genus distance. So in this case, we might assign this sequence as either being ambiguous between Enterobacteriaceae and Aeronesiae because its parent is ambiguous between these two. And the reason for that, as you can see, is that it has a children both as Aeronesiae and a child as Enterobacteriaceae. And so what we've done is we've tested Paratha using V1, V2, and V4, V5 on multiple different data sets. We've done it on synthetic data, comparing against data 2, which uses naive Bayes along with exact sequence matching for species identification. We've also tested it on mock communities here and real microbiome communities. In general, what we find is that it tends to assign more species assignments than data 2 while maintaining a fairly similar precision rate. And so this is a great news for us because it means we might be able to assign more species to a data set than we previously could. If you have more questions about its performance, I'd be happy to answer them. Finally, the last thing I'd like to point out is that because Paratha directly places sequences into phylogenetic trees, we can actually plot out where the sequence took place or was placed in, and we can get an understanding of why a sequence received the assignment that it got. So in this case, we can see we have a placement here, and it's near genera for both Trichoceus as well as Aerosphaeriae. And so we can see that Paratha wasn't confident in sounding a genus level because its distance was beyond the genus level threshold, which makes sense because its placement is actually at a part in the tree where two different genera appear. And with that, again, if anyone has any questions, my contact information is in the poster information section, and I'd be happy to chat more about Paratha. Have a good one. Thanks.